The topic is pharmacokinetic modelling, non-compartmental analysis versus population pharmacokinetics. I'll talk about some other things as well, just because I'm interested in them. I was told to make the talk friendly. I did all my slides and realised I didn't have a friendly picture, so I put one in. So this is one of the kids who was involved in the very first drug trial I was involved in when I started my PhD. So look, some of what's in this talk is my opinion, and I accept that my opinion changes over time, and I can be wrong as well, so I'm happy to be challenged on anything, and if I say something completely different next year, that's because my opinion's changed. There you go, that's my, I'm free now, my disclaimer. So I'll just talk a little bit about pharmacokinetics and probably echo a lot of what Darren had said earlier in the day, talk about why we care about it or why we potentially care about it, and then just go through some stuff about different ways of analysing pharmacokinetics, ending with an interesting example of using population PK. So this is what I feel my colleagues see when I use the word pharmacokinetics. And to be honest, I'm not going to have any of these in the talk today, thankfully. I'll have more trouble trying to explain them. Here's some pharmacokinetic terms, and Darren's already introduced some of these today, so I won't go really in depth. I'm happy to talk to anyone about anything about PK after today. But you know, we think ADME, absorption, distribution, metabolism, and elimination. And when we kind of look to examine the pharmacokinetics, often we can't differentiate between metabolism and elimination, and so we use clearance in our models for that term. With regards to distribution, that's more about the volumes, both the central volume and the peripheral volumes, and absorption is absorption. That one doesn't get a special different name. So this is one way of thinking about pharmacotherapy. We give a drug and something happens and we get a result. Either one that we're after, so efficacy, or one that we aren't really looking for, which is toxicity. And many of the drugs that we use in adults anyway, we use a fixed dose. But between giving that fixed dose of a drug and the result, there's a lot of potential covariates, and size is a big one. Age, there may be some changes in fat distribution, other things, disease states, organ function, genetic polymorphism, and usually even doing the most simple thing, which is adjusting for size. The pediatricians have got their heads around it. Adult physicians certainly don't, and anything that you got a dose milligram per kilogram, you call the pharmacist first. So in terms of day-to-day work and being an immunology reg, we hardly know how medications work, let alone dosing them in a very intelligent way yet. So there's two black boxes, this is one way I think about it, between the drug and the result. And the first one is pharmacokinetics, what we do to the drug, and the second is pharmacodynamics, what the drug does to us, or in the case of antimicrobial therapy, in terms of efficacy, we were interested in what it does to the bugs. Looking at pharmacokinetics, there's useful reasons to know more about pharmacokinetics of drugs. And this includes, before you even dose the person, that you have some information about how the drug is going to likely behave in them based on some population experience. And you say, well, they're male, they're this age, they've got this genetic polymorphism, we should start with this dose. And then after you start the drug, you get into therapeutic drug monitoring, and you can measure the concentration or a PD measure, and we all measure INR, so we do this. Everyone's doing this all the time anyway, technically. And then you adjust on the dose. But to be able to do that, you need to know that there is a target. So the concentration, the peak, the AUC, something about the exposure relates to the outcome that you're interested in, whether that's an efficacy outcome or a toxicity outcome. Okay, so here is a model for drug disposition, and this is time, x-axis, and concentration, y-axis. On the top, it's both linear and then a log linear plot on the bottom. And you can say, well, this is a real sample, so there's probably first-order elimination there, one-compartment model, and obviously an oral drug, so an absorption. And so this is my best effort to draw a human, a box. And that's how the models see human beings. They see them as a box. And that's, we call that box the volume, and that's the distribution part of the ADME. We then have to put the drug in, and that's my best effort of drawing a drug. And it goes in with an absorption rate into the box, and it gets diluted out to whatever volume it is. So the bigger the box, the lower the concentration, the smaller box, the higher the concentration. And in oral drugs and IV drugs, that relates to the peak. And then we get rid of the drug. Either we metabolize it or we eliminate it, and we call that clearance in our models. And that's really the slope down, how quickly we can reduce the concentrations. So that's probably one of the simplest models. The more simple one would be where you don't have to absorb the drug, you just kind of drop it in, give a stat IV dose. Here is another model for drug disposition, and this one obviously is far more complex. And you can see the drug itself gets absorbed and then it gets eliminated, but it's got two different metabolites. Itself has two different peripheral volumes, and then one of the metabolites has a peripheral volume, and the metabolites get eliminated as well, and it's really, really complex. And so we're going to start by saying all models are wrong, because the human being, we're complex, we've got different compartments, every organ will take up a drug in a different way, even some cells take it up, some cells don't. And so we're going to agree with George Box, at least the first part of what he said, and say that all models are wrong. And so that's where non-compartmental modeling comes in. Don't need to make any assumption about the underlying model of the drug, how it's absorbed, how it's eliminated, whether there's peripheral compartments or anything else like that. You can still compute key pharmacokinetic values for an individual, things that we're familiar with, so Cmax, maximum concentration, Tmax, the time at which that occurs, the area under the curve, and then via different mechanisms, the clearance and the volume as well. And also these other things which are becoming, they're hard to explain and not very useful, area under the first moment curve and mean residence time. It's good to describe what you see. That's what non-compartment model is good for, in terms of what you observe for that patient at that time. But it does become problematic when you have more sparse data, very limited ability to kind of extrapolate, and you can identify covariates in populations, but it becomes more difficult. So I thought I'll just talk you through an example of non-compartmental modeling. Once again, time on the x-axis, concentration on the right, and this is the same plot that we saw before, real plot. So we want to know what the maximum concentration is, so we just find the highest point, we draw a line that way, that gives us our Cmax, and we draw a line this way, and that gives us our Tmax. Easy as, sorted. Then, but you can appreciate that if you hadn't taken that sample exactly at 1 hour, and you're doing a whole bunch of people, you took some maybe 20 minutes too late, 5 minutes too early, that you may not be able to then compare between those people. So if you're doing studies like this, you need the samples from all the participants to pretty much be at the same time for all the points. And then for AUC, you draw triangles and trapezoids, and then you multiply them out. You figure out what the area is between all of those, and you get the area under the curve from 0 to your last time point. But you haven't got rid of the drug yet because you can still measure it at that time point. So this is when you flick to the semi-log plot, and you draw, I think that's pretty straight from there. I'll include at least 3 points. In this case, I reckon 7, and that's freehand. That's pretty much the elimination. And so using that, I can draw another triangle, figure out what the area under the curve is from 6 hours out to infinity, using figuring out what the elimination rate is, or the half-life. And then I add that to the area from 0 to 6 hours, and I've got my AUC from 0 to infinity. And those are the 3 things which the FDA looks for if you're doing a bioequivalence study. They want to know that the two drugs Cmax that AUC to the last time point and AUC to infinity are all the same. And so non-compartmental modeling certainly has a role. If you're doing a bioequivalence study, you've made a generic, you want to have a go at the big boys, and you have to prove that your drug within 90% confidence fits between 0.8 and 1.25 for those three parameters. So there is a role for non-compartmental. But problems arise, and this is actually busulfan in an oral busulfan in an adult, where only sparser sampling has been taken. And you can kind of guess what the problem might be, is that if you did the AUC here, then you would miss that top little bit there. And so you're not getting a full picture. And the only way to make sure you don't miss things like that is to take lots and lots of samples. And often in these studies where they're proving bioequivalence, there's between 12 and 15 samples up until the last time point. So it's not imminently helpful for when you are trying to do these samples in remote areas, in hospitals, in sick patients, to kind of get this full profile to make it, to give you the answers that you want. So to the second part of that sentence, all models are wrong, but some are useful. And this is my favorite part of that sentence because I apply it all the time. Some are useful, but they're all wrong. So compartmental analysis, you have to make some kind of assumption about the underlying model, the structure of the model. And while you're building the model, you have to assess that you're not introducing significant bias by introducing by the model that you've chosen. And often you need to go through a series of them and different absorption rates, different number of compartments, whether it's linear first order elimination or whether there's some Michaelis-Menten elimination. And so you can test as many as you like, basically, but you want one that doesn't introduce bias, hopefully. You can then analyze each of your patients individually, then summarize and try to identify covariates that link with your different parameters. But you still need quite a lot of data points for each patient. And to kind of demonstrate that, this is the one data point that I have for this patient on this trial. They were given valdecoxib during surgery, and then they got lots of breast milk samples, but they only bled them once. Can't really do anything with that one sample from that one patient. Luckily, she wasn't the only person on that trial, and everyone else on the trial got one sample as well. And so they all are contributing to the model, even though they are all individuals. And so we can't do compartmental modeling on each person individually, but maybe we can do something if we took the whole population together. There's some information there that we can extract. And so this is where population pharmacokinetic analysis comes in and is relevant for our discussion today in those populations where we can't always get the perfect number of samples that would allow us to analyze each person individually. So it allows you to use data from flexible sampling. Not everyone needs to contribute the same number of data points at the exact same time. That being said, you still need a good spread of data from where you expect the key parameters to be. So you can't just all collect them all 2 days later and they're all, half of them you can't detect and half you can. So having a spread, because each will contribute a certain degree to the estimation of, say, clearance or volume, and so you still need some points around those key areas. You can create really complex models, as complex as your imagination with boxes and lines and differential equations can get, or you could keep it simple. And sometimes you might find that the structure of the model might help you understand something about the drug itself. So if you're testing models with the whole population, you can say that linear elimination did not work and the model had less bias and was statistically had a better fit when we used Michaelis-Menten elimination. That tells you something about the drug. But not always. Sometimes nothing in particular. There is a structured way in which you can identify biologically plausible relevant covariates. You can include biologically irrelevant, unplausible covariates if you like, but they're less helpful. And in studies where, in your particular study, you studied a drug in a population that someone else has studied, but you quite didn't have enough information to estimate, say, clearance well or absorption well, you can incorporate prior information if you feel it's relevant, and then have that weighted into the final analysis as well. The thing I like most about it is that it enables you to do simulations. Of course, anything that you extrapolate from your population is extrapolation, and so you always take with a pinch of salt. But it can be helpful, as in the example with ARCO, in finding out, well, how high would we expect the Cmax to be? Has that Cmax been reported in the literature previously? Are we concerned enough that we should definitely not go ahead, or is this something that is likely to be okay? So how it works is probably not what I'm telling you, but this is kind of what the mechanics or the process is. I'm not mathematical enough to explain the algorithms that it uses to minimize all these things, but this is what it does. It takes all the data and analyzes it all at once. And in this case, for this particular drug, any points taken up that curve, it's telling you something about the absorption in the population. Around the peak is telling you something about the volume in that population, and then the downslope is telling you about the clearance. And so you have your population parameters. They may be affected by certain covariates such as size, renal function, perhaps drug interactions, other things. And then you have the first layer of variability, which is the variability between subjects. And that can exist, or you can test it for each of the parameters in your model. So you can have the population value for Ka, absorption rate, and then a variability around that as well. And you can estimate what that is. And the same for volume distribution, and the same for clearance. If you've given lots of doses, you can also estimate what the between dose variability is for those parameters which you think might vary from dose to dose, such as relative bioavailability between doses. The next layer of variability, you might call it error or residual unexplained variability. This is kind of what it looks like once you get it out, and here are 16 individuals. You can see that the black line is what the population average is for this drug at that dose, and so using the median population parameters. And then each individual has some variation from that curve based on their own parameters. And this is what the between subject variability is estimating. How different is the absorption rate between them? And you can see, if I can get this to work, which nope. Number 10 up there probably has a slower absorption because the peak is kind of over that way. Number 1 probably has a smaller volume, maybe a faster clearance. And to kind of demonstrate that more, I've taken one out of here. So this is the population curve based on the median estimates for parameters in the model. And then the individual curve is accounting for the between subject variability in this particular participant. The difference between those is due to the between subject variability, and although it's not normally distributed, normally log normal distribution is often or more likely, but imagine that the population value for volume distribution and clearance are in the middle there, then this person probably has volume distribution on that side of the curve and a clearance on this side. Then the difference between the observed points and the individually predicted line is what we're more used to, is this idea of residual error or residual unexplained variability. And that's not for for that individual, that's for each of those data points in that model. So here's an example that I chose, which was a model where we kind of pushed it a bit far you might argue, but we still think we got useful information out of it. So Lawrence comes to me and says, hey Sam, I want to know about meningeal inflammation and concentrations of Artemether after IM dosing, because we give it to patients regardless of, we can't differentiate whether they've got meningitis or cerebral malaria, so they get treated anyway. And we're worried that they're getting too much in their CSF based on the numbers that they saw. So the challenge, there's a number of challenges here. So just to explain the graph there. So the yellow dots are plasma in children without meningeal inflammation. And the yellow triangles are their CSF levels. Every child only got one paired sample. The blue dots are then those children with meningeal irritation. I think it was defined as more than 20 white cells per millimeter cubed. And then the triangles are their CSF concentrations. Now, the times for this were determined by clinical care. We couldn't mandate when they were supposed to have a CSF. And certainly we couldn't take, well, they couldn't take multiple CSFs, which is what I would have liked. It would be helpful in this case if it was possible to have more plasma samples to help build that model, but there are some good literature models on the disposition of IM Artemether in a similar population if we needed to use prior data to help support our current model. And a lot of the CSF samples were actually below the limit of quantification, so potentially not giving much information to us at all. There was also weights were missing, and in children I subscribe to using allometric scaling a priori. And so for me, it's a key covariate, and it was missing in 30% of the population. So you say, super dodgy, whatever answer you get is probably wrong, but there are things that we could do to work around this and see if there is any useful information in terms of quantifying this risk. So with regards to only having one blood sample and one CSF sample from each child, the limitation is that then you can't differentiate between what is between subject variability and what is residual unexplained variability, because you only have one sample for each child. The solution is, or one solution is, that you fix the residual unexplained variability, and you fix it to a reasonable value and something around that you might expect around the limit of quantification of your assay, and so we fixed it at 10%. It really didn't make a difference what we fixed it at, and we went through a process to check that it didn't change. The key answer that we're after is how much does meningeal inflammation affect CSF concentration of Artemether? So that's the question. The times were determined by clinical care, not by any protocol. So what we did was, at the end of it, we assessed whether this protocol was suitable in answering the very particular question that we had. And so we simulated out 1,000 populations, both with the same times and also the same number of samples below the limit of quantification, to see how good it would be to estimate the real value based on these simulations. For the samples which were below limit of quantification, instead of fixing them to a value which adds bias, or getting rid of them, which also adds bias, there is a method within non-Mehrmann and population pharmacokinetics where you flag those, and instead of trying to make the model fit those, instead you determine the probability that those samples are above the limit of quantification. There's no information. The information is that it was not detected by the assay, which has this limit of quantification, and this is the residual error in the model. And finally, for the missing weights, we used a multiple imputation method using the distribution of weight based on who weight for age tables as well. So I spent more time checking the result than I did actually running the model of 30 samples. And this is the answer that we got. The answer was that in those with meningeal inflammation, the expected concentration of Artemether was 5 times higher than those without. And then we simulated based on the final model to determine potential toxicity based on in vivo cutoffs. And I understand that we're really getting away from what we've observed now, and it's really extrapolating, but it's within the limitations of what we were able to do. And this is what we came up with in terms of simulating out to hopefully have some useful. And the information then to the clinician is that it's very useful for you to differentiate between meningitis and cerebral malaria, because you're potentially exposing kids with with meningitis to higher than acceptable concentrations of artemether. And of course, it's often they don't get dosed 7 days in a row. So we really went to an extreme example for the simulations. In summary, there's lots of ways to analyze PK, and it really does depend on what your question is. If you've got a generic drug and you want to prove bioequivalence, you have to do non-compartmental methods. And it's good to, if you can, design the study at the outset and decide how you're going to analyze the data at the end. Population PK, as opposed to individual compartmental or non-compartmental methods, does allow better for these real-world situations where you can't take 14 samples of 4 mils taken within 1 minute of the appropriate sampling time. And simulation, but junk in equals junk out, and know the limitations of your data and don't try to over-extrapolate. So in that example, we weren't making a comment about what the pharmacokinetics of artemether after IM dosing were at all. It was probably not very good at estimating those parameters at all. We just had the question about the meningeal inflammation. And finally, simulations can be helpful if you're considering different dose regimens as well. Thank you, and happy for questions and to be challenged.